Good afternoon, everyone. Thank you for coming to Workshop 23, CFTR Processing, Trafficking, and Interactions of Misfolded CFTR Mutants. My name is Deborah Shalon from the University of North Carolina in Chapel Hill, and I'm hosting this session with my co-moderator, Estelle Cormé-Boyaca from The Ohio State University. Please feel free to enter any questions during the talks in the Q&A box over to the right, and we'll get to as many questions as we can during the live discussion after each talk. I know this is going to be a great workshop session, and I'm looking forward to the talks. To get things started, our first speaker is Sophia Camalio from the Biosystems Integrative Sciences Institute in Lisbon, Portugal. Here we go.
Hello, everyone. First of all, I would like to thank you the opportunity to be here presenting my work. I'm going to talk to you about the characterization of 4 rare CFTR trafficking mutants and their response to rescue strategies. There are no relationships to disclosure related to this presentation. About 2,100 alterations in CFTR gene have been described to date, being about 400 CF causing. This huge number of mutations have been distributed into these 7 classes presented here according to their molecular defects. Among these 2,000 mutations, only about 18% are characterized, and some of these mutations are very rare and are present in very few patients worldwide. And that's why it's so difficult to predict the outcome of the disease. But however, some of these mutations will probably respond to the already approved CFTR modulators. Here, we are going to talk about class 2 mutations mostly. These class 2 mutations are mutations that affect CFTR trafficking, leading to the absence of CFTR at the plasma membrane. Identification of small molecules that correct F508del defect have been developed, and here now in clinics we have 3 corrector drugs which are present here. So we have VX809, VX661, and VX445. All these 3 drugs are used in combination with this potentiator, gating potentiator, VX770. So we have about 40% of patients with CF that are homozygous for F508del CFTR mutations. Then we have 34% of patients with CF that are heterozygous for F508del, and for these patients there are 3 treatments approved. So we have Orkambi, composed by VX809 and VX770, Sindeku, composed by VX661 and the potentiator VX770, and finally Trikafta, the triple combination that was recently approved, which is composed by 2 correctors, VX661 and VX445, and a potentiator, VX770. Then we have 8% of patients with CF that have gating mutations or residual function mutations, and for these patients we have ivacaftor approved, the VX770. However, for about 18% of patients with other mutations, there are still no drugs available, and some of them may respond to the already approved CFTR modulators. And that's why it's so important to characterize these mutations. Here we are going to focus on these 4 mutations. 2 of them are not even listed in CFTR2, so they are quite rare, and the other 2 are present in very few patients worldwide. So here you can see a representation of where the mutations are localized in CFTR structure. We have W57G present in the lasso motif. Then we have R56OS present in NBD1. Then in the ICL4 we have H1079P. And finally, in the transmembrane segment 11, we have Q1100P. So our aim is to characterize the rescue of 4 rare putative class 2 mutations by modulators and by the knockdown of cellular targets that correct F508del CFTR plasma membrane traffic. So we started by producing cell lines expressing these mutants, and for this we used the lentiviral system in which we introduced the CFTR cDNA bearing each of the mutants into the genome of these CFB cells parental. Then we assessed the effect of modulators both in CFB and intestinal organoids. And finally, we performed an siRNA-based assay to assess the effect of the knockdown of cellular targets. So the first questions that we asked were if the mutations impair CFTR biogenesis and if they respond to the corrector combination. So to address these questions, we started by performing a Western blot using the CFB cells expressing the mutants in the presence and in the absence of the CFTR correctors. And we observed that in the absence of any corrector, we couldn't detect mature form of CFTR, as you can see in all of the mutants. We also observed that none of the variants responded to the treatment with VX661. However, in the presence of the VX661 plus the VX445, we could detect a small amount or some amount of mature CFTR in cells expressing H1079P and Q1100P, as you can see here. These results were validated in intestinal organoids from patients bearing these mutations, and for Q1100P, you can also observe here a band corresponding to the mature form of CFTR. After this, we performed a functional analysis, and we did this in intestinal organoids from patients bearing these mutations, and we performed the forskolin-induced swelling, which is a technique in which we measure the CFTR activity or rescue through the increase of organoid area. So as you can see, following the treatment with VX661 plus VX445, we can observe in organoids bearing Q1100P mutation an increase in organoid area, as you can see here in this video. We also could observe this swelling for the organoids expressing W57G, but this increase in organoid area came from this other mutation present in the other allele, the 3272 minus 26 A2G. This mutation is a well-characterized mutation that is a splicing mutation, and because of that, it has some wild-type transcripts, and that's why we can observe organoid swelling even only with the forskolin and in the presence of the VX770. Then, since we didn't have any patient materials for this H1079P mutation, we performed another functional analysis. We performed using chamber, and we observed that following the treatment with VX661 plus VX445, we could detect a lumen negative response when cells were stimulated with forskolin, and this is a typical CFTR response. So we conclude that PhysAssay and using chamber results show that VX661 plus VX445 rescue CFTR function in cells expressing the mutants H1079P and Q1100P. Then we wondered if the mechanisms through which these mutations are retained in DR are similar to those causing the retention of F508del CFTR. And for this, we selected some targets from the literature that, when knocked down, rescued F508del trafficking defect. And we performed an siRNA-based assay in which we coated plates with siRNAs. We see that the CFB cells expressing the mutants on top of this, and then after 48 hours, we performed a Western blot to assess the protein expression. Knockdown of selective hits led to the presence of the detection of some mature form of CFTR for cells expressing F508del CFTR. And among the other mutants, only for R56OS, we could detect some mature CFTR form. No mature CFTR form was observed for any of the other three mutants under study. So our results suggest that the folding defect caused by these four mutations is more severe than the one caused by F508del, or that the recognition of such defects relies on different players in the cell. Also, this highlights the importance to study these very rare CFTR mutations and to try to find novel therapeutic targets to try to rescue these mutations. Finally, we also asked if second site mutations would allow the escaping of CFTR variants from DR. So it is known that mutants proteins can be retained in DR through the exposure of these arginine-framed 3-peptide sequences, And also that substitution of these arginine residues by lysines allow F508del to escape from the air quality control and reach plasma membrane. So we wondered if we could introduce these 4RK mutations and if we could rescue the trafficking defect of these 4 mutants. So we inserted these 4RK mutations in Cs with the other mutants, and we performed the Western blot, but we observed that none of the mutations was rescued by the 4RK in Cs. And this suggests that the structural impact of these mutations in CFTR is distinct from the one caused by F508del. So as a summary, in the area of highly effective modulators, we still need to characterize the defect associated with each mutation, to test the response to approved drugs in cellular models and in materials from individuals with CF. And evidence is accumulating that even when the teratype is similar, as it happens here, so we have F508del and the other mutants, the cellular machinery involved may be different, prompting research into specific mechanisms. So I would like to thank all my colleagues and my supervisors and you for listening. And if you have any questions, just let me know. Thank you.
All right. Thank you for that, Sophia. It was a very interesting talk and perfect for this workshop as we're looking at folding mutations. And those are some rare ones, which are very interesting to learn about. I'm not seeing any questions. Estelle, do you see any? Estelle is behind the scenes, helping me go smoothly here.
No questions yet.
Okay. Well, audience members, feel free to ask. If you're a little bit shy, that's okay. You can post as anonymous. We'll never know who you are, but we'd be happy to discuss what's on your mind. And you never know, you might ask something and then say, oh, or somebody else might ask something. You might say, oh, I wanted to ask that. Oh, here I see a question. Whoops, where did it go? Oh, here. This is coming from a speaker here. Catherine Oliver is wondering, can you discuss how the plates are coded with siRNA?
So we do we have a protocol in our lab in which we coat the siRNAs with a mixture of the transfection reagents and sucrose, and then we put the plates in the speed vac, so that we have all the liquid evaporated, and then we can save these plates to use later, and after and when we want, we can see the cells on top of these siRNAs already coated with the transfection reagent. It's easier way to do this since we have a lot of siRNAs, and it's easier than to do a liquid transfection.
Very nice, thank you. Now I'm seeing on Slido from Lars Plate. The question is, did I see correctly that R560S band B levels decrease with the corrector combination? Have you followed up to see what might be going on and whether the correctors destabilize the protein?
Thank you. We actually didn't do any more experiments. We want to know, we are more interested in rescuing this mutant because we are studying for some time now. And we cannot find a way to, or a compound to rescue it. We can see some of rescue with the siRNAs, with some of the siRNAs, but it's very modest. So it's a great question, but we haven't done it.
Very good, thank you for the question and the reply. And we have a question from Guido Veit, who's here as a workshop speaker. His question is, does the drug response in organoids translate into clinical benefit in patients?
So we have the only response with in organoids for the mutation H1079P and Q1100P. Since we didn't have any organoids with this H1079P, we cannot know. We only tested in CFP cells, but for Q1100P, we are testing. We tested and we can see some benefits with the triple combination.
Very good. That triple combination, super helpful.
Yes.
We have another question here from Anthony Sinadinos. Maybe naive experimental question. My former lab head used to say there are no simple questions. It's better to ask. Anyway, do your Western antibodies or antibody target all variant CFTRs equally?
So for organoids, I don't know if the question is for organoids, Western blots, or CFB cells, but we use a mixture of CFTR antibodies so that we can cover all we can really have the signal, most of the signal that we want. So I think we can detect all kinds of CFTR.
Very good. And here's a question from Justin Chin. Are there any plans to characterize the rare organoids as monolayers using electrophysiology?
So in our lab, we also try to do these 2D monolayers and to try to do this using chamber assays in these 2D monolayers, but we haven't done this with these organoids. But it's good to try.
Definitely. And here's a question from Liwa He from here at UNC. Question for Sophia. How does the 4RK decrease the level of B band of the mutants?
So the 4RK mutation, the 4RK studies that we performed were performed in HEK cells. So this can be due to the transfection efficiency. So I don't think we cannot, we can say that we have low band B.
Very nice. I actually have a question. Oh, sorry. I'm sorry, I cut you off.
I was just saying that we cannot know since it's transfection, transient transfection. That was just it.
Makes sense. I had a question with your second site suppressors. Why do you think there wasn't any enhancement of mutant CFTR folding?
I mean, probably the defect that is caused by these mutations are more severe than the one that is caused in F508del, in which we have these motifs that are that when mutated with the 4RK mutation are like hidden, and then F508del can escape. In these mutants, maybe the structure of CFTR is too like destroyed or altered, so that even the 4RK mutations cannot mask this defect in the structure.
Very nice. That makes sense. Well, I think that's about it for questions. So we can go on to our next speaker, which is Catherine Oliver from Emory University.
Hello. I'd like to begin with thanking Drs. Estelle Cormet-Boyaca and Deborah Shalon for the opportunity to present this project entitled Discovery of Novel Epistatic Interactions that Influence CFTR Folding Trajectory. Highlights of findings presented here are also available at poster 614. My disclosures for this presentation include funding from CFF and NIH, as well as a scientific collaboration with Ionis Pharmaceuticals. Before presenting the work, I'd like to acknowledge collaborators who have contributed to this project, specifically highlighting members of the Hartman Laboratory at UAB, Drs. Mert Ickes and Sean Santos, as well as Hee-Young Mao from Dr. Eric Sorcer's laboratory at Emory. In the era of CFTR modulators, patients receiving FDA-approved pharmacologic agents have exhibited robust improvement in clinical endpoints. However, additional effective therapeutic interventions are still necessary, as significant fractions of the CF population are unresponsive to available small molecules, experience adverse events, and/or carry refractory CFTR variants ineligible for modulator therapy. To address unmet medical needs, our work focuses on understanding genetic interactions that influence biogenesis of both common and rare CFTR defects. In collaboration with John Hartman's lab at UAB, we have utilized a novel approach known as genome-wide yeast phenomic analyses to identify genetic modifiers of mutant CFTR processing. Yeast express an ABC transporter homologous to CFTR, termed YOR1, or yeast oligomycin resistance 1, which functions as an efflux pump that extrudes a mitochondrial toxin, oligomycin, across the cell membrane. As you can see, this protein possesses a tertiary structure very similar to CFTR. A mutation analogous to the common delta F508 variant in CFTR was introduced into YOR1 at phenylalanine 670, leading to protein instability, misfolding, endoplasmic reticulum retention, and diminished function, which strongly resembles the delta F CFTR abnormality in mammalian cells. The YOR1 delta F670 variant was crossed with 5,000 plus single gene deletions from the yeast deletion strain library, and the resulting haploid double mutants were assessed for enhanced resistance to oligomycin. In other words, we were interested in those gene deletions that could rescue or overcome the delta F folding defect. Representative plates of delta F haploid double mutants are shown, where isolates were plated on growth media, or YPEG, and YPEG supplemented with oligomycin. Examples of colonies that survived in the presence of oligomycin are indicated in red. We identified several gene deletions that would be expected to improve delta F processing, such as enzymes involved in ubiquitination, like UBC13. But we were surprised to find that deletion of a ribosomal gene, RPL12A, resulted in robust restoration of delta F-mediated oligomycin resistance. Therefore, RPL12 disruption appeared to rescue functional expression of the delta F CFTR equivalent in yeast. I'd like to just mention that other ribosomal proteins emerged as hits, and we are following up on those leads as well. To briefly introduce RPL12, it is a component of the 60S ribosomal subunit and forms a base of a flexible structure called the P-stalk that is shown here. RPL12 is also a constituent of the GTPase-associated center, or GAC, which can interface with any GTP-bound translation factor. Based on the literature, RPL12 has an established role during elongation through its interactions with elongation factor 2. But due to its location in the GAC, this ribosomal protein likely plays a role in other aspects of translation as well. RPL12 also performs extra ribosomal functions, as has been described for a number of ribosomal proteins to date. In the interest of time, I would like to summarize an extensive amount of previous work that confirmed genetic interactions identified from yeast phenomic screening are conserved between yeast and the human airway epithelium, at least insofar as CFTR processing is concerned. Following siRNA-mediated suppression of RPL12, we found enhancement of delta F CFTR maturation efficiency, interdomain assembly, plasma membrane localization, thermal stability, open channel probability, and transepithelial ion transport. These findings were confirmed in CFBE, FRT, and/or primary human bronchial epithelia homozygous for the delta F variant. This work also included a number of collaborations that are shown here with collaborators at UAB and Utrecht. In collaboration with Zoya Ignatova at the University of Hamburg, we utilized ribosome profiling coupled to RNA-seq, otherwise known as RiboSeq, to assess global and transcript-specific changes in translation dynamics following RPL12 knockdown. We observed that RPL12 silencing slows initiation and elongation, primarily through effects on GC-rich codons, pairing to high-abundance tRNAs. In turn, this resulted in distinct changes to the CFTR translational profile, with reduced ribosome velocity at specific residues in the first transmembrane domain, R domain, and second nucleotide binding domain. In the present study, we investigated whether other genetic factors could either augment or inhibit the RPL12 effect on delta F CFTR biogenesis. A second yeast phenomic screen was performed, as before, using a YOR1 double mutant harboring the delta F variant and RPL12A deletion. This construct was crossed with approximately 5,000 single gene deletion mutants, and resulting colonies were assessed for increased or decreased resistance to oligomycin. Heat maps on the right display interaction scores for subsets of genes that differentially impact functional expression of the delta F RPL12 double mutant, or control strains such as the delta F variant alone, shown in blue, the YOR1 null, shown in red, or the YOR1 null with RPL12A deleted, shown in orange. From these data, we detected genes that promote delta F processing in the presence or absence of RPL12A deletion, also assessing whether target suppression affected delta F with some degree of specificity, meaning without impacting phenotype of the YOR1 null strains. For example, genes that enhance delta F biogenesis independent of RPL12A deletion are enriched, as evidenced by a reduced L score, shown here, which is the cell proliferation parameter representing the time at which maximum growth occurs. We were most interested in genes that only augment delta F biogenesis in combination with RPL12A deletion, as shown here. Functional enrichment of several targets from this subset are annotated on the right, such as factors involved in translation initiation and GPI anchor remodeling, as well as additional ribosomal proteins. This list also includes genes of interest that inhibit delta F processing in the context of RPL12A suppression, such as EMC2, a component of the ER membrane complex. In order to assess relevance of yeast findings in the setting of the mammalian airway epithelium, we performed siRNA-dependent suppression of gene targets in FRT cells stably transduced to express delta F CFTR, harboring an HRP tag in the fourth extracellular loop. Genes of interest were silenced alone or in combination with RPL12 knockdown in high throughput fashion. Cell surface localization was quantified by addition of an HRP substrate, and resulting luminescence was normalized to live cells. The latter were detected using a Promega cell titer fluoride viability assay, in which live cells uptake a fluorogenic substrate that is cleaved by a protease. Results from the first subset of genes evaluated are plotted here, with single gene depletion shown on the left and double gene depletion shown on the right. Please note that NT refers to non-treated and NS refers to the non-specific siRNA control. Black asterisks annotate statistical comparison to the non-specific siRNA, whereas red asterisks annotate statistical comparison to the RPL12 knockdown alone. As you can see, silencing RPL12 as a single agent increases delta F trafficking to the same degree as treatment with the corrector VX661. Additivity between RPL12 siRNA and concomitant knockdown of PGAP2, which is a GPI anchor remodeling factor, is also apparent. In contrast, EMC2 suppression exhibits inhibition of the RPL12 effect, which is congruent with findings in yeast presented earlier. The second group of genes tested are shown here, which largely encompass constituents of the ribosome. Interestingly, RPL21 silencing rescued delta F CFTR cell surface localization to similar levels achieved by RPL12 knockdown or application of VX661. Synergistic enhancement of delta F trafficking was observed when suppression of RPL12 was combined with inhibition of RPL21. In addition, suppression of RPS11 alone modestly augmented delta F plasma membrane density, but together with RPL12 knockdown of these two genes also appeared synergistic. Since I cannot discuss each gene of interest in detail, I would just like to highlight RPL21, as suppression of this target exhibited the most robust epistatic interaction with RPL12 inhibition. RPL21, otherwise known as EL21, it resides in the 60S subunit and has a primary role in ribosome assembly in the form of pre-rRNA processing, as well as cytoplasmic translation. I should also note that EL21 does not interface with RPL12, shown in red, nor the GTPase-associated center or the peptidyl transferase center. However, a recent study from the Dalian group suggests that RPL21 possesses an extra ribosomal function in regulating transcriptional expression and proteasomal degradation of the protein lysine methyltransferase SMID2. Silencing RPL21 could exert effects on delta F CFTR through any of these interactions, and because the pathways are distinct from those impacted by RPL12 knockdown, perhaps this is why we observe synergy with dual suppression of the two targets. Investigations are underway to explore a number of mechanistic hypotheses that are related. A question that invariably arises when I present the RPL12 story pertains to off-target effects of RPL12 suppression. We have assessed a number of parameters that I will just briefly highlight here. In yeast, RPL12A deletion restores mutant UR1 function but does not affect the wild-type UR1 processing. No evidence of cytotoxicity has been observed to date in FRT, CFBE, or primary airway epithelia following siRPL12 knockdown, as judged by a number of different assays. Expression patterns of CFTR-associated chaperones listed here are unaffected by RPL12 inhibition. RPL12 knockdown does not exert global effects on trafficking of other polytopic membrane proteins, either native or mutant glycoforms with ER retention phenotypes, similar to delta F, and those are provided later. In addition, RPL12 heterozygous mice are haplosufficient. Both sexes exhibit normal weight, growth, lifespan, reproduction, and no adverse phenotypes detected to date. So please note that specificity of silencing additional genes of interest, such as RPL21, will be evaluated in similar fashion in the future. To summarize, genome-wide yeast phenomic analyses led to the discovery that RPL12 suppression rescues delta F CFTR functional expression by reducing translation velocity, thereby allowing additional time for the ribosome and associated chaperones to confer a more native-like protein conformation. In this study, yeast hits that were specifically epistatic to RPL12A deletion represent the largest gene set, suggesting RPL12 may have a primary regulatory role in delta F protein processing. Epistatic interactors include genes involved in lipid remodeling, phosphatidylcholine homeostasis, ER-associated protein folding, translation initiation, and components of the ADS ribosome. In FRT, siRNA-mediated knockdown of RPL12, together with some yeast targets, augments delta F CFTR plasma membrane density in additive or synergistic fashion. Ongoing studies include examining whether dual suppression of these targets leads to enhancement of delta F CFTR mRNA abundance or stability, protein maturational efficiency, thermal or proteolytic stability, transepithelial ion transport, and so on. In conclusion, our findings describe new epistatic modulators of CFTR folding, which may also serve as therapeutic targets to further enhance RPL12-mediated correction of the delta F variant. This study emphasizes the power of utilizing unbiased, high-throughput yeast phenomics to discover novel factors influencing a genetic disease, such as CF. I'd like to end by announcing that our pediatric research enterprise at Emory and Children's Healthcare of Atlanta is growing. We are recruiting basic clinical and/or translational investigators with an immediate opening for a mucosal immunologist described here. Furthermore, my own laboratory is hiring students and postdocs. If you are interested in either of these opportunities, please do not hesitate to contact me via email. Thank you for your attention, and I would be happy to take questions.
All right. Thank you so much for that cool talk, Catherine. First, I didn't realize that your one yeast homologue has such structural similarity to CFTR. That certainly comes in handy. So first, we have a question here from Margarita Amaral. Do you think the effect of RPL12A deletion could be related to giving more time for delta F508 CFTR to be synthesized and or folded?
Yes, I think that's one of the prevailing hypotheses because with the slowing of initiation and elongation that occurs with RPL12 depletion, that's what we hypothesize, is that we're just allowing more time for interdomain contacts and native-like conformations to form. And we also see, for example, when we combine RPL12 knockdown with low temperature correction, that there appears to be no degree of additivity or synergy. So it's potentially overlapping mechanisms there.
Nice. Next, we have, again from Portugal, it's from Carlos Farina. To Catherine, number one, does silencing of these ribosomal proteins have an effect on overall translation rates? And two, how do you envisage targeting these proteins therapeutically?
Yes, so we have thoroughly interrogated the effects of RPL12 depletion on translation rates, and the overall speed of elongation is decreased from around 5 amino acids per second to about 2 amino acids per second. The initiation is also decreased, but that's not a rate per se, but I can just say it's several fold down. And so we intend to utilize similar mechanistic investigations that we've done in the past, employing ribosome profiling, for example, to determine how inhibiting RPL21 or other targets from these screens might impact translation kinetics. And then the second question, can you repeat what he said there?
Sure. How do you envisage targeting these proteins therapeutically?
Yeah, so there are, I guess, a couple of different methods one might pursue. The first one being, small molecule inhibitors. But another one that I mentioned as part of my disclosures is I'm working with Ionis Pharmaceuticals to develop antisense oligonucleotides against these proteins. And with the vision being that, transient depletion of ribosomal proteins should not, be as impactful as a lentiviral-based or an adenoviral-based intervention. We wouldn't have to necessarily worry about repeat administration effects. So I think my favorite right now is the ASOs, but yeah, there are other ways too.
Nice, and it's great using RPL12 as a target, as in one of your final slides you showed, it doesn't have any global effects or negative side effects, if you will. So that is fantastic, because I would think, if you knock out a ribosomal protein, bad things are going to happen, but you've looked at that and it doesn't. So that is really great and promising. Next, there's a question from Lars Plate. He's wondering, the increased dependency on the EMC with RPL12A knockdown is interesting. Besides membrane insertion, the EMC is also proposed to have chaperone functions for membrane proteins. Do you think the function is particularly important here to stabilize intermediates during the translational slowdown?
Yes, I would say that's one possibility. And the other one being that, other components, or I guess other targets besides EMC2 that were additive or synergistic with RPL12 included factors that they inhibit the ER-associated degradation pathway. So in essence, by doing that, we're theoretically increasing the pool of misfolded CFTR, or in other words, correctable CFTR, where we can add something like an RPL12 suppression intervention, or maybe even small molecules such as a Trikafta-like treatment that might be additive or synergistic together to rescue biogenesis of CFTR. So I would say that there's probably combinatorial effects of, like, depending on what the role of that particular EMC factor is. It might be deleterious for CFTR folding, or it might be enhancing for CFTR folding if you suppress it. So.
Very nice. Those are the questions I see so far. Are there any more? I see another one. This is from my co-moderator, Estelle. How does the rescue compare to Trikafta? You had 661 alone, so how about triple, or at least two correctors?
That is something that is under testing right now. So I'll be able to tell you at next year's NACFC.
That's fantastic. We anxiously await that. All right, so if there are no further questions, we'll move on to our third speaker, who is Min-Soo Kim from Vanderbilt University. Here we go.
Good afternoon, and thank you for joining my talk. My name is Min-Soo Kim, and I'm a third-year graduate student in the Plata Lab at Vanderbilt University. My talk today is titled Elexacaftor, or VX445-mediated CFTR interactomics remodeling of misfolding mutation. Before I begin, I would like to disclose no relationships related to this presentation. Cystic fibrosis is a lethal genetic disease that mainly impacts the lungs, where mucus buildup obstructs airflow and becomes prone to infection. CF occurs due to loss of function mutations in the cystic fibrosis transmembrane conductance regulator, or the CFTR protein. Close conformation structure is shown on the right, where CFTR is a cyclic AMP regulated anion channel that helps regulate fluid transport to clear airways. CFTR biogenesis is tightly regulated, and mutations can impact functional CFTR production at multiple stages and are classified as so. Class 1 mutants are terminated prematurely, whereas class 3 and 4 involve gating and conductance malfunction. We study class 2 mutations that entail misfolding of CFTR and subsequent premature proteasomal degradation. Some example mutants are listed here. Several small molecules have been discovered as pharmacological chaperones that help stabilize the structure of CFTR to avoid this premature degradation, thus restoring the amount of mature CFTR on the cell surface that can be functional. Shown are drugs currently being used in the clinic. Ivacaftor is the first approved corrector drug for the F508 deletion mutation. Below is its analog tezacaftor. Elexacaftor on the bottom is a second-generation corrector recently approved with better outcomes than previous correctors. But these drugs were initially approved for the most common mutation, F508 deletion. We explored other rarer mutations and how they respond to these drugs. We do this through immunoblotting for CFTR. Wild-type CFTR shows up on a Western blot with a thick C-band of mature post-Golgi CFTR and core glycosylated form of CFTR as the B-band. However, for mutants such as F508 deletion and P67L, the C-band is lacking. Interestingly, drugs like VX809 can restore the C-band to varying degrees, as shown. It is puzzling why the response to VX809 is inconsistent across mutations. As you can see in the structure, P67L is an N-terminal mutation at the last motif, whereas delta F508 is on the nucleotide binding domain 1 in the transmembrane domain 1 interface. We could perhaps attribute the differential response to the structural defects that manifest in unique ways in each mutant that interfere with drug binding. The drug could also influence critical protein-protein interactions that occur during CFTR biogenesis. We have explored VX809 differential response in a preprint cited below. Furthermore, the recently approved drug elexacaftor also shows variability in correction. Another hyperresponsive mutation, L26W, which is located near P67L in transmembrane domain 1, responds well to VX809, as well as VX445, as indicated by the increase in C-band from no drug treatment. Interestingly, P67L does not respond to VX445. Notice how VX809 restores C-band levels to almost wild-type levels, but VX445 has a faint band that's comparable to DMSO. This is again shown in this Western blot quantification bar graph, where control and VX445 treated P67L C-band were not statistically different. A similar trend was reported by Veit et al. As well, where VX445 showed minimal correction of P67L. We've also observed this result through Using chamber experiments done by the Sorcerer group, where FRT cells stably expressing P67L were treated with either VX661 or VX445 or both. VX661 conditions showed channel function almost back up to wild-type levels, whereas VX445 at 3 micromolar did not show much improvement and did not show a dose-dependent response at a higher concentration, 5 micromolar. VX445 is clearly ineffective on P67L CFTR. As mentioned, CFTR biogenesis is highly regulated at multiple stages by protein homeostasis factors or proteostasis factors, starting from translation, translocation, to folding machinery, chaperones like HSP70s and 90s. Once misfolded, degradation machinery such as E3 ubiquitin ligases come in, where ubiquitinated CFTR are degraded by the proteasome. Properly folded CFTR can then be trafficked to the Golgi for processing and to the plasma membrane, where it can be then either recycled or endocytosed for degradation by the lysosome. The interactome of wild-type CFTR and several mutants, including delta F508, have been published by Pankow et al., have shown how different these profiles could be. Since mutations impact protein-protein interactions, it is likely that small molecules that can counteract these mutations are to reshape the interactions as well. For instance, interacting proteins may be altered by the drug, which leads to differential correction of CFTR, either binding directly to these interactors or at the interface between CFTR and the interactor. In fact, Hutt et al. Has shown how VX809 restores delta F508 protein interactions toward wild type. Therefore, we could learn from mapping the interactomics profile of mutant CFTR in response to drug treatment, and it could help us understand the biological mechanism of these drugs, especially when not a lot is known about the second generation corrector, elexacaftor. So far, I have shown that VX445 does not restore mature P67L on Western blot, electrophysiology, and similar trends were observed by the Lukács group. This prompted us to question, why does VX445 show varying amounts of correction? To answer this research question, we decided to investigate protein homeostasis contributions to the differential drug response. This is an overview of our approach to identify CFTR interactors to map the proteostasis interactions. We employ tandem mass spectrometry to ID interaction proteins, filter and group identified interactors into pathways, and compare directly altered pathways by drug treatment, then pinpoint proteins and validate them. We identified interactors by performing multiplexed co-immunoprecipitation. We transfected HEC293 T cells with CFTR mutant constructs and drugged them accordingly. Then cells were lysed and immunoprecipitated using CFTR-specific antibody conjugated beads to obtain CFTR interactors. Then we digested and labeled the peptides with tandem mass tags to multiplex all conditions into a single quantification for throughput. From MS2, mass reporter tags are fragmented. They can be deconvoluted to directly compare intensities across conditions in a quantitative manner. With the interacting proteins identified, we compared them against the control transfection to filter out true interactors. An example of volcano plot here is shown where log 2-fold change of the tandem mass tag reporter ion intensity is shown on the x-axis, and p-values are shown on the y-axis. We filtered for interactors that are at least one standard deviation away from the log 2 normal distribution of all interactors. And this was done on each condition to generate a master list of true interactors for further analysis. Next, we compared each mutant with and without drug treatment over the wild type to observe aberrant interaction behaviors. Shown here is a hierarchical clustering of interactors grouped based on intensity similarity, where each bar is an interacting protein, and yellow color indicates increased interaction with CFTR when compared to wild type. Notice how P67L mutant overall has increased interactions that are potentially triggered by misfolding of the protein and recruitment of proteostasis factors. With VX809, these interactions can then be toned down. The bars are darker than DMSO, whereas VX445 profile resembles that of DMSO interactions. For example, I'm highlighting the green and blue clusters where this trend is evident. Similarly, in L26W, without drugs, there are increased interactions. These interactions are decreased with VX809 and VX445. Notice how in the purple cluster, there's a group of interactions that only VX445 was able to remove for L26W. This stresses how these correctors can uniquely influence protein-protein interactions. To further analyze which pathways in P67L are being differentially impacted, we classified each of these interactors into cellular pathways. And these pathways are relevant with CFTR biogenesis, such as folding, trafficking, and degradation. With this grouping, we ran statistical analysis where two pathways stood out as significantly altered by VX809, but not VX445. Shown here is a violin plot with individual data points as each interactor protein classified into the respective pathway. The degradation machinery showed a decrease in abundance with VX809 treatment, but not with VX445. We can speculate that corrected CFTR will likely not encounter degradation machinery once they're properly folded. Interestingly, a stronger trend was seen in the translation machinery, where again, we observed a decrease in abundance with VX809 treatment, but not with VX445. This could be due to increased dwell time in the endoplasmic reticulum of CFTR due to misfolding, which may increase the likelihood of CFTR interacting with translation-related proteins in the ER. We have also seen the same pattern for these two pathways in our preprint for VX809 and P67L in completely separate experiments which adds validity to our results. Lastly, we did a direct comparison of VX445 over VX809 to line up the interactors from high to low interactions in an unbiased pathway-free approach. The waterfall plot here shows lined up interactors, and as expected, several proteins from the two disrupted pathways, translation and degradation, were ranked high and stood out as targets for validation via knockdown studies. Our future work includes siRNA knockdown of candidates and pathway inhibition with proteostasis regulators. By knocking down these interactions, we expect to observe some rescue of P67L and VX445-induced rescue of P67L. To summarize our findings, we have shown that VX445 is not effective for P67L. Protein interactions are more abundant with mutants than with wild type, where effective correctors can mitigate these interactions, but ineffective ones do not. VX445 has implications as early as translation and degradation when it comes to P67L correction. Our result is the first TMT multiplexed interactomics data on elexacaftor and its effect on CFTR. Understanding the CFTR proteostasis network and how correctors influence it may reveal the cellular mechanism of correctors and hint how we may address unresponsive mutants. I'd like to acknowledge the Plata group and the Sorsha group, Eli and Caroline especially, for working on the Cystic Fibrosis Drug Interactomics project, Disha and Andras for the electrophysiology data. Also, I'd like to thank the NIH and the Cystic Fibrosis Foundation for their support with funding. Thank you for listening.
All right, Min-Soo, thank you for a great talk. Let's see, are there any questions? I'm not seeing any yet. Feel free to type those into our Q&A box. Anyone? Don't be shy. Oh, I see some coming in. Terrific. To Min-Soo, this is from Carlos Farina. This is Portugal representing today. In your comparisons, mutants show a lot of increased interactions, but very few decreased interactions. Wouldn't you expect some decrease in interactions, for example, with late trafficking machinery?
It's a really good question. I've been thinking a lot about that recently. So since mutants that are well corrected with, for instance, a drug, are more likely to make it to this plasma membrane and therefore should be interacting more so with the proteins that are post-Golgi, like cytoskeleton proteins. We did not observe that. And one of the reasons that we think is that these mutants are, since they're misfolded, they're innately very sticky and are prone to interacting with proteins in the vicinity in general, and therefore they're shown to have increased interactions. They're also, the way we treat or process our samples, we try to capture the interactions at steady state. So we keep our cells in ice and try to preserve the protein-protein interactions while we're preparing them for our mass spec runs. And during the process, it is possible for the cells or the proteins to form additional interactions, which then leads back to my first point that they're sticky and therefore maybe forming additional interactions in addition to what they initially had. And so, yeah, that's my explanation, but I'm not really sure.
Very nice. Always nice to hear speakers give an explanation even if we don't really know. There's always something to think about. So thank you for the question and the answer. Anyone else? I actually have a question. So now that we know that P67L does not respond to VX445, it makes me think about Trikafta versus Symdeko. I can't remember off the top of my head. Are patients with P67L approved for Trikafta or Symdeko? Because I would imagine that 661 would be beneficial I think it is beneficial, just like VX809. So the issue is VX445. So in thinking about what medications these patients would get, if there's no point, if 445 is not doing anything to correct the protein, there's no point in these patients being on Trikafta. So I'm just wondering if they are approved, and sometimes there's a disconnect with what clinicians want to prescribe versus what researchers actually know on the molecular level.
Yeah. So Trikafta wasn't initially approved for P67L, but recently, like several months ago, they expanded the list of approved mutations, and P67L was on there. And as I've shown on my Western blots, VX661 is fully capable of increasing P67L CFTR mature levels that got almost up to the wild type. And sure. I would like to point out that the cell lines that we've been using aren't necessarily not as, I guess, significant as CFB cells. And some other research groups have shown that there is a little bit of correction by 445 on P67L in other cell types or cell models. And therefore, I think this added, albeit slightly, still better for patients.
Excellent, thank you. And there's a question that came in from John Hunt. Duchenne's data show that VX809 combined with VX661 bind essentially immediately adjacent to the P67 site, suggesting it directly offsets destabilization of that region in the protein. Do you see evidence that P67L doesn't just correct that local folding defect and prevent interaction with proteostasis factors?
I'm gonna have to reread this question, and I'm having a hard time formulating.
Are you able to see it and read it? Sometimes it
yes.
helps to process while reading, sure. Just making sure you can see it.
So yeah. So based on our method, we can't really distinguish whether or not this change in interactors are based off of purely the structural correction that these correctors are providing, or whether or not these interactors somehow may actually influence the way these drugs bind to CFTR. So we're unclear on that. But yeah, it's an interesting point. But so far, based on what I have seen throughout my research, I think that it's probably the structural changes that that follow these correctors binding that are then influencing downstream interactors. I hope I answered the question.
Very nice. Something to think about. If not, I'm sure John Hunt will chime in again. And we have a question from one of our speakers, Guido Veit. Which cell model was used for the proteomics study? Which percentage of the interactors is expressed in primary HBE?
So we used HEC293 T cells. And I'm not sure what percentage of the interactors are expressed in bronchial epithelial cell lines. So yeah. And yeah, we would like to actually go, because the previous interactome studies done by other groups, they all used CFBE cells for delta F508. However, these primary cell lines aren't available for these rare mutants. And well, I guess one of the speakers, Sophia, actually made their own CFBE cells by transfecting with viral means, and I think it'd be interesting to actually move over to more relevant cell lines in our lab, and we're kind of pushing for it at the moment.
It's good you have that in mind, but it's still fine to test in other cell lines. Don't let them make you feel inferior. And coming up next is Guido Veit from McGill University, and he's going to be talking to us about P67L. Here we go.
I would like to thank the organizers for the opportunity to present my data. The title of my talk is Optimized Modulator Combinations for Rare CFTR Mutants with High Responsiveness to Single Correctors. I have no disclosures. As you all know, the modulator combination drug Trikafta, which combines the folding correctors Vertex 445, Vertex 661, and the potentiator Vertex 770, provides unprecedented clinical benefit for CF patients with 1 or 2 copies of the delta F508 mutation. Importantly, the addition of one drug, Vertex 445, or also called elexacaftor, almost triples the correction efficacy for delta F508. This is shown here by biochemical analysis and also by functional measurement in HBE cells. Consistent with this, the addition of Vertex 445 to the tezacaftor I combination yielded an almost tripling of the delta PP FEV1 effect in the clinical trials. Our first question was, what is the efficacy of Trikafta on an absolute scale? For this purpose, we measured the functional correction of CFTR in homozygous delta F508 human nasal epithelia from 5 patients in comparison to the CFTR current in H&E from 10 wild-type donors. When we combined the 2 correctors, Vertex 661 and Vertex 445, and acutely added the potentiator Vertex 770, we achieved a correction of the current equivalent to about 80% of the wild type. When the cells were chronically exposed to Vertex 770, the functional correction efficacy was reduced to approximately 65% of the wild type. This is still multiple times the correction efficacy of first-generation correctors in combination with chronic Vertex 770. Therefore, we wanted to know what is the mechanism of Vertex 445. We and others have previously shown that different correctors target distinct domains in misfolded delta F508 CFTR. Type 1 correctors such as Vertex 809 or Vertex 661 bind to the MSD1 and stabilize the interface between the membrane spelling domains and NBD1. Type 2 correctors, such as corrector 4A or other aminothiazole compounds, bind to the NBD2 and stabilize this domain and the interactions to other domains. For a long time, there was no highly efficacious type 3 corrector which would stabilize the NBD1 directly. In 2018, we were able to isolate one compound, 4172, which binds to the NBD1 directly and is highly synergistic with type 1 and type 2 correctors. To test whether Vertex 445 follows a similar mechanism, we performed 2 assays. First, we determined the spontaneous aggregation of the NBD1 domain with a delta F508 mutation. This can be inhibited by the addition of glycerol or inhibited by the addition of a bacterial chaperone system. Vertex 445 attenuated the aggregation of the delta F508 NBD1. In the second assay, we measured the loss of the secondary structure of the NBD1 domain by CD spectroscopy. Again, this loss of secondary structure is attenuated by Vertex 445, indicating that Vertex 445 directly binds to and changes the unfolding trajectory of the delta F NBD1 domain. Next, we analyzed the efficacy of the combination of Vertex 661 and 445 for the correction of rare folding mutations located throughout the channel. In many cases, substantial correction was obtained that exceeded the efficacies of single compounds, regardless of the mutation localization. This indicates the allosteric function of the corrector molecules, which lead to an overall stabilization of the channel. This is consistent with the functional correction of rare CFTR folding mutants in patient-derived H&E cells. Here we found that the combination of vertex 661 plus 445 corrected the function of rare mutants to about 20 to 40% of that of the wild type. This is a clinically relevant correction level. However, Trikafta vertex 445 does not only act as a corrector, but also as a potentiator for delta F508 and gating mutants such as the G551D. Acute addition of elexacaftor increases the activated and vertex 770 potentiated current of both delta F508 and G551D, indicating that vertex 445 acts as a co-potentiator. In summary, we and others have shown that elexacaftor with tezacaftor and ivacaftor corrects the functional expression defect of a variety of CFTR mutants with folding defects. Elexacaftor can act also as a potentiator together with ivacaftor and corrects the gating defect of delta F508, gating and conductance mutants. This is consistent with the recent approval of Trikafta for additionally more than 170 CFTR mutations located throughout the channel, based on in vitro data in FRT cells. So this leads to the question, should every patient with a folding and gating mutation receive Trikafta? To answer this question, we have to look at drug-drug interaction, adverse drug-drug interaction, that make it desirable to administer as few CFTR modulators as necessary. These adverse drug-drug interaction include that chronic ivacaftor treatment reduces the efficacy of the correctors lumacaftor, tezacaftor, and the combination out of elexacaftor and tezacaftor for the correction of delta 508 and rare folding mutants. Another adverse drug-drug interaction is that vertex 809 induces the metabolizing enzyme of vertex 770 and thus reduces the vertex 770 plasma concentration. To answer the question whether some mutants require a reduced number of CFTR modulators for maximal functional correction, we first looked at the relative correction efficacy of vertex 661 and 445. We found that there's a cluster of mutations in the membrane spanning domain 1, which are more readily corrected by vertex 661 in comparison to vertex 445. We picked two of these mutations, P67L and L206W, as well as a mutation in the NBD1 S549R, which is associated with a mild folding defect, for further analysis. Biochemical analysis showed that these mutations are very well corrected by type 1 correctors such as vertex 809 or vertex 661, and that addition of vertex 445 provided only moderate additional benefit. P67L and L206W are also associated with a peripheral stability defect. These defects were corrected by either vertex 809 or vertex 661, and again, the addition of vertex 445 provided only very little additional benefit. The mutations P67L and L206W are often considered to lead to a mild form of cystic fibrosis. However, if we follow the percent predicted FEV1 in patients with these mutations over time, there's at least a subset of patients which experience progressive loss of their lung function. We were able to obtain nasal epithelia from all of these patients to determine the efficacy of modulator combinations to rescue these mutants. H&E carrying one allele of the L206W mutation and the delta F508 or splicing mutation on the second allele responded very well to type 1 correctors. However, vertex 770 potentiation did not provide additional benefit. We also observed that there's a huge variability in the current of H&E derived from different patients. Importantly, there's a tight correlation between the basal current and the vertex 809 or vertex 661 corrected current, indicating that the amount of residual channel activity predicts the corrector of efficacy in individual H&E. To focus on the modulator effect of L206W, we were investigating the H&E cells with a splicing mutation on the second allele, which likely does not give rise to protein production. In one of the two patients, we found moderate benefit of vertex 445 as a second corrector. But again, there's no benefit of vertex 770 addition. Next, we determined the functional correction patient-derived nasal epithelia with one allele of P667L or S549R. For P67L, the combination vertex 661 plus vertex 445 provided benefit in comparison to single corrector treatment. However, chronic addition of vertex 770 did not increase the channel function. In contrast, an S549R required vertex 770 mediated potentiation. The vertex 770 potentiated current was further increased by type 1 corrector addition. However, addition of a second corrector, vertex 445, did not provide additional benefit. To summarize this, mutations in the membrane-spanning domain 1, including the P67L, E92K, and L206W, exhibit preferential response to type 1 correctors. A correction level comparable to Trikafta was achieved for S549R by vertex 661 plus vertex 770, and for P67L and L206W by 661 plus vertex 445. Thus, introduction of a third modulator may not provide additional benefit for patients with a subset of rare CFTR missense mutations. These results also suggest that H&E as a precision medicine model can be used to optimize mutation-specific modulator combinations. Finally, I would like to acknowledge the people who were involved in this project, and I'm looking forward to your questions. Thank you.
Right, Guido, that was an amazing talk. So much data, very interesting, so many mutations, so many treatments. Fantastic. Do we have any questions? Once again, don't be shy. Questions for Dr. Veit. I'll just comment. It's more of a comment. Maybe it can become a question. So in thinking about your P67L data and then Minsu's P67L data, one thing is you did it in, those were nasal, I think in one of your last panels, it was in nasal cells, and because it's from a patient, of course, there's P67L, and there was also delta F508, which we know responds well to Trikafta. So it could be that those responses that we're seeing, particularly to acute VX770, that could be responses we're seeing from correction of delta F508, of course, right?
Yes, that's correct. Basically, these mutations are so rare that it's quite difficult to obtain homozygous nasal epithelial cells. And for this reason, the only cells which were available for us had a delta F508 mutation on the second allele.
course. So that could explain why it's why those patients are approved for Trikafta, because more likely than not, the other mutation is going to be delta F508, and then once you have that, you're approved for Trikafta. But I do wonder, side by side with I can't remember if you had it. Sorry, if you had just the components of Symdeko versus Trikafta, looking at that patient P67L delta F508. I know you had multiple combinations. I can't remember now if you had 661 with just acute 770, or I guess chronic 770.
Yes, so the addition of 445 as a second corrector provided benefit. So Symdeko actually is definitely worse than Trikafta, but again, the component ivacaftor in the Trikafta would probably not be necessary.
Yes, I agree with you 100% on the 770 component not being necessary. Here's something from John Hunt. Thank you. Is there any sign that the allosteric interaction patterns of VX770 are different from the correctors?
I'm not sure as in how far the effect of 770 is allosteric. I mean, okay. Let me rephrase that. The effect which we are seeing for 770 is, or the inhibition of the corrector effect mediated by 770 is really quite selective for a few mutations only. So far, we only have identified a handful mutation. It's definitely less than there are mutations responsive to correctors. So there's no clear overlap of these both effects.
All right, thank you for that. Any other questions? Here's one from Catherine Oliver, one of our workshop session speakers. Very elegant, Guido. For the delta F508 P67L genotype, have you tested more than one patient code and noticed any significant differences in modulator response?
We only had one patient code available, and but still I would expect that there are also differences in the response between individual patients, as we've seen for the L206W.
Very nice. So I had a question about the figure where you had VX445 acting as a potentiator. So you may know that the Gench and Alexandrov labs are also interested in this. And if you look at poster 616, where Andrei Alexandrov is looking at VX445 by single channel measurements, there's also some Ussing chamber data there where we used bronchial epithelial cells. And it was a delta F508 patient, and similar to what you've done, we added acutely in Ussing chambers, first VX445 and then VX770, and then the opposite, first VX770 and then VX445. So the thinking that we have is because, like you've shown, VX445 assists with folding, assists with processing, that's the first step. So first VX445 creates more available channels, and then VX770 acts on those corrected channels as a true potentiator. So in our studies, we saw that when we first add VX445 and then VX770, that there was enhanced channel activity versus in the other sequence where you added first 770 and then VX445. And I think your data showed the opposite, where when you first add VX770, I think of it as then enhancing the function of VX445. So if it is acting as a dual corrector and potentiator, that maybe it's being prompted or amplified somehow by VX770. So I don't know if that's really a question or more just up for discussion and thinking about, I guess, in different systems, different patients, they may respond differently. And also just in thinking about whether VX445 actually is acting as a potentiator or just acting as a corrector to create more channels that a potentiator could then alter the open probability.
Okay,
So.
That's a very interesting question. Thank you for asking that. Yes, indeed, we have seen actually the opposite, that Vertex 770 is permissive for the acute action of Vertex 445. Interestingly, what we've also seen is the EC50 for the potentiator function of vertex 445 is about 100-fold lower than the corrector EC50, which may suggest that there are two different mechanisms and possibly also two different binding sites for vertex 445. And some data which we just recently obtained in the lab is we were trying vertex 445 in a construct which was lacking the NBD2 domain. So in a classical model, we would assume we get R domain phosphorylation, then NBD1, NBD2 dimerization, and then channel pore opening. However, in this model where we lack the NBD2 domain, we still see an co-potentiator effect of vertex 445, which may suggest that 770 plus 445 in combination achieve pore opening with possibly without NBD1, NBD2 dimerization.
That is very interesting. Thank you for providing feedback on that observation / question. And I see another question that came in from Lee Cody. Is it possible then that some patients may do worse on Trikafta?
We have not found evidence for that, but obviously from a simple pharmacological perspective, I think it would be beneficial to have as little drugs as possible, since there may be all kind of drug-drug interaction, particularly since patients have to take these drugs lifelong.
Yes, absolutely. And also considering that we know that VX770 accumulates in tissues and serum and everything. So yeah, if there's a way that the channel can be corrected, inserted into the membrane, but doesn't need the extra kick of potentiation that VX770 provides, that'd be super. All right, any additional questions? Last call for questions for Dr. Veit. All right, and our final speaker in this workshop session is Frédéric Anglès from Scripps Research.
Good afternoon. My name is Frédéric Anglès from the Balch lab at Scripps Research, and today I'm going to talk about the impact of genetic variation on CFTR trafficking and how physical and chemical correctors are impacting CFTR trafficking. Before starting, I would like to thank the organizer for the possibility of presenting my work today. And I also would like to thank the Cystic Fibrosis Foundation that has financed my work for the last 3 years. So cystic fibrosis is a recessive monogenic disorder that's caused by mutation in the CFTR gene that code for the CFTR protein. CFTR is a chloride channel localized at the plasma membrane of epithelial cells, and it's an ABC transporter composed of two transmembrane domains, TMD1 and TMD2, that are each composed of 6 transmembrane regions. You have two nucleotide binding domains, NBD1 and NBD2, that are going to interact with ATP, and a regulatory domain. In the cell, wild-type CFTR is translated in the ER and go through the exocytic pathway to reach the plasma membrane. It goes through the Golgi where it's glycosylated, and on the Western blots for wild-type, you can actually see two bands appearing, band C and band B. The band C is the glycosylated form that went out of the ER and through the Golgi, and the band B is what is remaining in the ER. In the patient population, we observe that the most frequent variant is the F508del variant, the deletion of the phenylalanine at the position 508. And this variant destabilizes the protein in the ER, and it's nearly not exported. So on the Western blots for the 508del, you see mostly band B and nearly no band C. For each variant, we can calculate a trafficking index that represents the capacity of a variant to be exported. And to do that, you calculate the ratio between band C and the total band C plus B. In the case of the wild type, the trafficking index is nearly 1, as mostly everything is in band C form, while for the 508, nearly everything is in the band B form, so the trafficking index is close to 0. In addition of the 508, more than 2,000 mutations has been observed in the population, and nearly 50% of them are predicted to be missense mutation. In our lab, we have a library of 70 different variants that are localized a bit everywhere on the structure, and those missense variants are causing cystic fibrosis. And when you calculate the trafficking index relative to wild type for all of those variants, you see that some variants are impacting the trafficking index, while others are actually not impacting at all the trafficking of the protein. So in order to understand how any missense mutation would impact the property of CFTR, the lab has developed a few years ago a new Gaussian process regression method based on geostatistics. It's called variation spatial profiling. It's used a sparse collection of data points to predict the property of an entire landscape based on the localization of those data points. Using the same approach, we can use all the variants as data points, their location, and their impact on the trafficking to actually calculate which region are most likely to be important for the trafficking of the protein. If we gray out all the regions that have at least 50% trafficking compared to wild type, and we focus on the really defective region, we can see that only TM1, 2, and 3, 11 of the transmembrane domain 1 and 2 are mostly impacted. The ICL4 intracellular loop 4 of the TMD2 and most of the NBD1 domain. All of those regions are the ones that are most likely being essential for the trafficking of the protein. Now that I know that, I was interested in looking how decreasing the temperature would impact the entire CFTR fold. It's been known for nearly 30 years now that the 508 is actually temperature sensitive. And by decreasing from 37 to 26, for example, the temperature, you can improve the trafficking index of the 508 compared to the DMSO 37 degree. But it's been 30 years and nearly no work has been done on other variants. And we don't know yet what temperature decrease is doing to the entire CFTR fold. So what I did first is I look at the trafficking index at 37 or 27 for the library of the 70 variant we have in the lab. And so focusing on the variant that display a defect in trafficking, you can see that actually most of the variants are not responding to the temperature decrease, and only few of them show a real difference between the 37 and the 27 degree for the trafficking index. And for example, we have the 508 that is highly responding to 27 compared to 37, but we have most of the other variant like the S492F that is actually not responding at all at 27 compared to 37, even if the S492 is really close to the F508 in the MBD1 domain. So I calculated the delta between the trafficking index at 37 and 27 for all the variants and apply VSP to decipher the impact of temperature on the entire protein. And what's interesting is that calculating the delta, you see nearly no difference between 27 and 37 for most of the protein. And only one particular region is really corrected when decreasing the temperature. And this region is the region at the interface between the ICL4 from the TMD2 domain and the nucleotide binding domain, a specific region of the nucleotide binding domain. When you have the F508 here, the R560, what's interesting in this region is that it's centered around the YKDAD motif, which is the export motif, has been shown to be essential for the export of CFTR. This motif is recognized by COP2 at the ER membrane, and this is the cargo export motif that is essential. And so basically what we show here is that reduced temperature stabilize interdomain NBD1 ICL4 and improve deacidic exit motif recognition by COP2, but does not act on any other part of the protein. Then I looked at the impact of the new correctors, elexacaftor and the combination Trikafta on the CFTR trafficking using the same approach. So I looked at the impact of Elexa or Trikafta compared to DMSO for all of the 70 variants we have in the lab, and focusing on, again, on the variant that display a defect in trafficking, we see on the opposite that actually most of the variants are responding, but still some of them are not responding. And so we have variants that are not responding at all to either Alexa or Trikafta. We have some variants that are not responding to Alexa compared to DMSO, but the Trikafta is improving the DMSO. We have other variants where it's the opposite, while Alexa has a huge impact on the variant, but Trikafta doesn't add anything on it. And finally, we have specific variant where Alexa has a huge impact, and adding another corrector in the combination of Trikafta is improving even more that correction. So using the same approach, the data on all of those variants and the trafficking index after treatment with Alexa or Trikafta, I used VSP and looked at the impact of Alexa and Trikafta on the entire CFTR structure. And what you see is, compared to DMSO, you see an improvement of most of the transmembrane region after Alexa treatment and a mild effect on the ICL4. And a small part of the MBD1. Adding the other corrector in the Trikafta combination completely stabilized most of the variation in CFTR. You see nearly no defect anymore in the transmembrane region. The ICL4 is mostly corrected, but interestingly, we still see a specific location where we don't see any correction. And when you look specifically into it, you observe that actually the major core not being corrected by Trikafta is the YKDAD motif, the export motif, that is not corrected at all. So any mutation happening here is not corrected by Trikafta. And if you see, you can see a first layer around that core that can be corrected, but it's still a mild correction compared to the rest of the protein, meaning that those variants in the first layer are also impacting a bit the core, and all of this region is actually not being corrected by Trikafta. So I looked at the stability of 508 in HBE cells stably expressing 508. And I looked at 37, 27 after treatment with Alexa or treatment with Trikafta. And so in the control 37 DMSO, you see only BNB that is rapidly degraded after 4 hours when you use cyclohexamide that's going to block the translation. And you see 4 hours later, nearly no BNB. In the case of 27 degree, you see a decrease of BNB and a decrease of BNC. So the corrected BNC is also not stable and is degraded nearly completely after 4 hours. And there is a correlation between BNB and BNC. So basically what it means is that the 27 degree is improving the export of the 508, but not its stability per se. On the other end, Alexa and Trikafta, when you treat the cell with those and you look at the stability, you see that nearly no destabilization of BNC compared to the disappearance of BNB, meaning that here, what has been exported and reached the plasma membrane is actually extremely stable compared to the correction that we see here with the 27 degree. And interestingly, when you actually combine those two corrections, the Trikafta and the 27 degree, you see a huge improvement, 5 to 10-fold compared to the Trikafta alone at 37, and of course, to the DMSO control at 37. So what I show here is basically that the Trikafta stabilized globally the structure of the protein, and basically any missense mutation impacting the stability can be corrected by Trikafta. However, if you are impacting the export motif and the export itself of the protein, you're not being corrected. On the other end, the temperature shift mostly improve the export of the variant that are impacting the deacidic exit motif and nearly doesn't do anything for the other variants. And put together, you have a huge synergistic effect between the two different types of correction that are either impacting the stability, consequently improving the export, but also the export itself. And combining the two is how you can actually rescue 508 to nearly a wild type level. So just to give you a quick peek of what I've been doing recently is like I showed that the Trikafta properly stabilize nearly the entire fold of the protein. And what we are missing to really correct entirely the 508 is an improve in the export itself. So I've been looking into the COP2 system. It's composed of around 12 different proteins with different isoforms. And it's recruited at the ER, is interacting with CFTR and gonna form different layers that gonna form the vesicle and go through the Golgi. This is the trafficking and the export of CFTR. And I looked into the modulation of those components. And I've seen that modulating specific component of the COP2 system can actually improve CFTR export. I looked into the interactome of those specific COP2 components and the proteome in HBE cell upon silencing of those components. And combining all of those data, we actually find that some specific COP2 direct and indirect pathway can improve a lot CFTR export. And combining that with the current stabilization, you can actually rescue nearly to wild type level the 508 mutation. And so with that, I would like to thank again the Cystic Fibrosis Foundation that has financed that work. I would like to thank Dr. Phil Thomas at UT Southwestern that has provided generously the library of plasmid that was the foundation of my work. I would like to thank Professor Balch and Professor Hutt that has been my two mentors for the last 6 years. I would like to thank Dr. Chao Wang, who developed the VSP approach in the lab, and all the other lab members. Thank you very much for your attention.
All right. Well, thank you, Frederic, for that amazing talk. And I see there's a question from John Hunt from Dr. Veit's talk about VX445. I think I'll get to that after. Any questions for Frederic, just to keep things moving along with questions and then that are grouped together with the speaker that just spoke. So feel free if you have any questions for Frederic, just submit them anytime, please. Just wait a few seconds here. I know it's the end of the day. Here we go. John Hunt is back. It's great questions. Thank you for those. John Hunt asks, there are earlier reports that VX809 does not stabilize CFTR residency in the plasma membrane. Is VX661 fundamentally different, or are the earlier studies wrong?
Could you repeat that? Sorry, you take me by surprise.
Sure. Also, you might see it in the chat box if it helps a little to absorb it by reading, but I will read it again happily. Again, it's from John Hunt. There are earlier reports that VX809 does not stabilize CFTR residency in the plasma membrane. Is VX661 fundamentally different, or are the earlier studies wrong?
So I didn't look into the VX809 per se for all the mutants in the lab. I actually look only into elexacaftor and the Trikafta combination. So I'm not sure I can really talk about the single effect of just one drug because I always looked at them in combination, actually. The only drug I looked was the the VX445. And what I've seen is a huge stabilization at the plasma membrane. In the ER, I actually don't really know how the stabilization occurs. What I can say is that because we've seen a huge improvement in export, I can hypothesize a good stabilization in the ER that's actually decrease the degradation and so improve the export per se. The stabilization by VX661 and 809 I actually cannot really say per se.
All right, well, thank you. And there's a question here from Anthony Sinadinos. Starts with silly one. I guess he thinks it's a silly one. And again, there are no silly questions, but it's an interesting idea. Has anyone tried cold air inhalation therapy? Of course, this isn't thinking about low temperature. That's not really comfortable for humans, but maybe a little bit colder might help.
This this is I don't know, but that's interesting because it's a joke that Professor Balch does all the time about just we need to put the kids in the Michigan lake and they would be fine, but obviously we cannot do that. But yeah, so the idea using the temperature was to find a way to understand how it impacts the entire structure and what are the key important regions that are rescued by the temperature effect. So we would actually learn more about the pathway and the different means of correcting 508 and other mutations based on the observation of the rescue by the temperature.
Very nice. And another question from Margarita Amaral to Frederic. Do you find that several COP2 machinery are additive among themselves?
Additive? Actually, no. So basically, I silenced all the COP2 components and all the different isoforms, and we've done some additive silencing, and we've seen actually only 2 different components that are really improving the export. And we didn't see much of an additive effect of those 2 components, meaning they are probably impacting the same pathway within the COP2 machinery when we silence them to improve CFTR export.
All right. And Margarita wants to add, by the way, great talk. John Hunt comes back with, I may have misread your data. So you didn't examine VX661 without VX445?
No, we did VX445 alone. So basically the idea came a year and a half ago. And when the data started to come in with the VX445 and Trikafta, and the 661 data, 809 as well, were previously done by several groups. So because we have a certain amount of variance, the replicates, doing all the experiment, I actually minimized the number of conditions I would have. So I did not replicate all the data that was done on 809 or 661 alone, and just look at 445 alone because no one have done that before, and at least for a library in this size, and the Trikafta combination.
Very nice. Thank you for that dialogue that we had through digital and through live speaker. Any other questions for Frederic?
Just to further comment on the last question, actually, this is still something we have been looking at using the VSP approach. It's just we did not generate the data ourselves, but we actually used the same approach to look at the impact of the 661, 809 alone. And we see definitely a different type of rescue between the 661 and the 445, for example. And we do see a lot of differences on the impact on the structure. I didn't show that, I didn't have the time, and we did not produce the data ourselves. But using those available data and the method we have developed, we actually looked at it, and it's in a paper we recently submitted.
Excellent. And John Hunt would like to add, thanks for the clarification, my misunderstanding. This is why we have discussions. Fantastic. Any other questions for Frederic? You can still have a few minutes here in the meantime. Also, does anyone have questions that they thought of for the other speakers? While you think about that, I'll go back to John Hunt's question for Guido Veit about VX445. John Hunt asks, isn't the VX445 action, maybe this is more for me, but we can discuss it together, Guido. Isn't the 445 action in the electrophysiological experiments basically instantaneous, meaning that activity can't be secondary to correction, making the discrepancy in Andre's data more mysterious?
So maybe I can comment a bit on that. If I understood the poster correctly, the hypothesis is that Vertex 445 would enhance phosphorylation of CFTR and thus provide more channels which are pre-activated, thus 770 could have a bigger instantaneous effect. As I said, we have maybe some data which contradicts that, but I think that's definitely for a mechanistic point of view, still an ongoing study since we haven't done single channel measurements. I may also add that it would make a lot of sense to rather use the G551D since the potentiator effect of 445 in macroscopic measurements is so much bigger in G551D compared to delta F508. And also we have some data for wild type showing that we don't have any acute effect of 445 in wild type.
Yeah, G551D would definitely be good to look at. I saw you looked at that and I thought, oh, that would be helpful for us. I think in our poster, again, 616, if you want to talk to Andre, please feel free. We use delta F508, delta F508 homozygous, and in the bronchial airway epithelial cells. Otherwise, the single channel measurements had delta F508 or wild type. For these studies with VX445, I don't think he looked at G551D, or those data were not conclusive. But in the poster, the single channel recordings again are delta F508 and wild type. And yeah, G551D would definitely be useful. And getting back to John Hunt's question, yeah, it's instantaneous in the Ussing chambers. So if you add VX445, even if it's acting in the processing, phosphorylation, if you see a response acutely in the Ussing chamber, then it has to be some sort of potentiator activity. So maybe that goes back to what you said, Guido, about perhaps there are multiple binding regions or multiple sites of activity, perhaps one for correction, one for potentiation by VX445 on the molecule. Maybe. All right, let's see if there's anything else. Did I miss any questions? I'm looking in multiple places. All right, any other questions for any of the speakers? All right, well, I'd like to thank all the speakers for their outstanding research and wonderful presentations. And thank you for all for coming and helping make this an interesting discussions with your great questions that prompted wonderful conversations. And here in the era of triple therapy, Trikafta, we all know that it's very important still to study issues revolved around CFTR processing and folding and interactions. I hope you all have a very nice day and enjoy the rest of NACFC 2021.